Hello students. Today we are going to perform the fourth practical of your biology syllabus, where we have to analyze a urine sample to check the presence of normal and abnormal constituents in it. So let us start by studying the aim and the principle for this experiment. The aim of this experiment is to test the presence of urea, sugar, albumin and bile salt in urine. So as we know, a normal urine is pale yellow in color. And this coloration is due to the pigment present that is called urochrome. The pH range of a normal urine lies between 4.8 to 8 and it contains about 95% of water and 5% of solutes like sodium, potassium, magnesium and ammonia. Now, if these solutes or any other normal organic compounds like urea, uric acid or creatinine is present in urine, they are considered to be the normal constituents of urine. But if constituents like sugars, albumin, bile pigments, blood or pus is present in urine, they are considered to be the abnormal constituents of urine. So as we know, the metabolic functioning of our body, it generates many products and byproducts. And these byproducts, they are removed from the body mainly through the fecal matter and the urine. And so urine analysis is routinely used for the diagnosis of number of metabolic disorders that can take place in a body. Now to perform this experiment, the first test is test for urea. Now let us see the requirements to perform this test. We require certain glasswares like beaker, test tubes, test tube holders, and then we require spirit lamp or a burner. The chemicals that we require are phenolphthalein indicator, urine sample and soybean flour. Now, let us also see the procedure for this test. We have to take a test tube, and in that test tube we have to add 2 ml of urine and a pinch of soybean flour and then mix it well. After that, we have to add about 1 to 2 drops of phenolphthalein indicator and keep it in a water bath for about 5 minutes. Now students, to understand this procedure even better, let us see the working part and then we'll discuss about the observation and the result. So students, let us start with the working part of this experiment. Now in this experiment, we are going to study the presence or absence of certain normal and abnormal constituents into a urine sample. Okay, so let us start with it. The first one is detection for urea. Now for that, you have to take urine sample into a test tube. So I am taking about 5 ml of urine sample into the test tube, and in this we have to add a pinch of soybean flour. So I am just adding a pinch of soybean flour and mix it well. After mixing it, we have to add phenolphthalein indicator. So about 2 to 3 drops of phenolphthalein indicator goes into this, and again we have to mix it well. And then we have to keep this test tube in a water bath for about 5 minutes. And after 5 minutes, if the color changes to pink coloration, that means urea is present into this urine sample. So let us see the result after 5 minutes. Okay. Now after keeping the test tube in the water bath for 5 minutes, you can already see the pink coloration in the test tube. Okay. This pink coloration indicates the presence of urea into the given urine sample. So here you can see a very clear pink coloration. That means urea is present in the given urine sample. Okay, so as you have seen the working part, where in the urine sample we added a pinch of soybean flour and then 1 to 2 drops of phenolphthalein indicator and placed it in a water bath for about 5 minutes. After 5 minutes, we saw that there is a pink coloration that is developed in the test tube. This pink coloration indicates that urea is present in the given urine sample. The soybean flour that we added, it provides an enzyme urease. This enzyme, it catalyzes urea to ammonia and carbon dioxide. Ammonia being alkaline, it reacts with phenolphthalein and it develops the pink coloration. Now urea, it is produced in liver as a normal catabolic process of excess amino acid. And this urea, it is filtered out by the kidney. And its presence in the urine, it indicates a normal functioning of the excretory system. Now if there is an increased amount of urea, the condition is called as uremia. And this high level of urea, it indicates too much of protein diet or higher protein breakdown in body. On the other hand, if there is a low level of urea, it indicates kidney problem or inadequate protein in diet. So students, this was about the first test, that is the test for urea. Now let us see the second test. The second test is test for sugars. And the chemical that we require is Benedict solution and of course the urine sample. Let us also see the procedure. We have to take 2 ml of urine in a test tube and add 1 ml of Benedict's reagent. So Benedict's reagent is the one that is used to determine sugars. After adding Benedict's reagent, we have to boil it for about 2 minutes. And then we have to see the color change. So now let us see how this test is performed and then we'll discuss the observation and the result. Okay, so now let us do the second test. In the second test, we are going to test the detection of sugars. Okay, so we are going to detect the presence or absence of sugar into this urine sample. So again, we'll take a clean test tube and in this we have to add about 5 ml, approximately 5 ml of urine sample. And to this, we'll add Benedict's reagent. So students, let me tell you, Benedict's reagent is the one that is used to detect the presence of sugar in any sample. Okay, so now you can see that the initial color for this is somewhat bluish green coloration. Now we have to heat it for 5 minutes, and after heating, if the color changes to yellowish orange coloration, that means sugar is present into this urine sample. So students, on adding Benedict's reagent into the urine sample, the initial color that we got was somewhat bluish green coloration. Now we will heat it for about 2 minutes and we will see the color change. If the color changes to yellowish orange color, that means glucose is present or sugar is present into the given urine sample. So now while heating it, you have to make sure that you do not continuously heat it. If you heat it continuously, what will happen is all the material, all the content inside the tube will just pop out. Okay, so we have to avoid such things. So we have to heat it very slowly, giving it some time in between. Okay, you can already see the color changing. So as you have seen the test performed, where in the urine sample we added Benedict's reagent. And after adding the Benedict's reagent, the initial color that we obtained was bluish green color in the test tube. And then after heating it for about 2 minutes, we obtained a yellowish orange color precipitate. This precipitate, it indicates the sugar is present in the given urine sample. And this is because the glucose reduces alkaline copper sulfate, which is present in the Benedict's reagent, to cuprous oxide. And that gives the orange precipitate. So sugar, it is present in the blood in the form of glucose. And its level is kept normal by a hormone called insulin. Now if there is a deficiency of insulin in the body, the glucose level will rise in the blood. And this glucose will find its way in the urine. This condition is called as diabetes mellitus. Now sugar, being a reducing agent, it reduces copper sulfate into cuprous oxide. And as per the quantity reduced, it gives different color precipitate. Now if there is no change in the color, that means after adding the Benedict's reagent and after heating it for about 2 minutes, the color still remains blue or green, that means sugar is absent. But if 0.5% of sugar is present, then the color will change to yellow. If 1% of sugar is present, it will give orange precipitate. And if 1.5% of sugar is present, it gives brick red precipitate. So in the test that we performed, we obtained orange precipitate that you can see in the second picture. That means there was 1% of sugar present in the urine sample that was provided. So students, after this, let us move to the third test, that is test for proteins. The chemicals that we require over here is urine sample and concentrated nitric acid. Let us see the procedure. We have to take 1 ml of nitric acid in a test tube, that is a concentrated one, and then we have to hold the test tube at a 45-degree angle. And in that, we have to add about 2 to 3 drops of urine sample from the side of the test tube. Now how it is done, let us see in the working part, and then we'll discuss about the observation and the result. Let us do the third test. The third test is test for proteins. We are going to detect the presence or absence of protein into this urine sample. So again, taking a clean test tube, we'll add nitric acid to it. So please remember, for the test of proteins, the first thing that we add in the test tube is nitric acid. So it is a dilute nitric acid that we have to add in the test tube, and then to this nitric acid we have to add the urine sample. Okay, so there is a way of adding urine sample to this nitric acid. You have to hold the test tube in a slant position, take few drops of the urine sample, and you have to add it very slowly. It has to slide through the test tube and go into the nitric acid. Now after adding few drops, if I hold it straight, I will see a white cloudy ring that is formed at the junction of the solution. If this white cloudy ring is appearing, that means protein is present into this urine sample. Okay, so this is the nitric acid ring test. So students, as you have seen in the test performed, there was a cloudy white ring that was formed in the test tube. And this cloudy white ring indicates that albumin is present, which reacted with nitric acid, forming a white precipitate in the form of a ring. And so this test is known as nitric acid ring test. Albumin in urine, it gets precipitated with nitric acid. And albumin is one of the protein which is found in blood. Now normally during urine formation, these large molecules of albumin, they never escape ultrafiltration. But if presence of albumin is detected, then it indicates damage of nephrons, as in nephritis or urinary tract infection. So students, after this, let us move to the last test, that is test for bile salt. Now the chemicals that are required to perform this test is urine sample and sulfur powder. And how do we perform this test? We have to take 3 ml of urine sample and then sprinkle a very little quantity of sulfur powder on the surface of the urine sample. And we have to see if the sulfur powder is sinking at the bottom or it is floating at the top of the urine sample. So let us see how it is performed, and then let us discuss about the observation and the result. Now students, let us do the fourth and the last test, that is test for bile salt. Okay, so we are going to detect the presence and absence of bile salt into this urine sample. So again, we will take a clean test tube, and in this test tube, we will add about 5 ml of urine sample. And to this urine sample, the next thing that we have to add is sulfur powder. Now let me tell you, when we add sulfur powder and if it sinks at the bottom very quickly, That means this urine sample is having the presence of bile salt. Okay. So in absence of bile salt, the sulfur powder actually floats at the top. But if the bile salt is present, it will sink down at the bottom. So let us see. Now I am just adding a pinch of sulfur powder. And you will see that the bile salt is settling at the bottom. Okay, that means this urine sample is having bile salt. So with this, students, we finish all the 4 tests for the detection of normal and abnormal constituents in the urine sample. In the next slides, I will be of course attaching the pictures where you can see the result very clearly. Okay. So now, as you have seen the observation of the test as well, where we saw that the powder, it sinks rapidly at the bottom, and this indicates that the bile salt is present in the given urine sample. Now bile salt, it actually reduces the surface tension of the urine, and because of which after adding the sulfur powder, it will sink at the bottom. And this test, it is termed as Hayes sulfur powder test. Now bile salt, it is a secretion that is produced by liver, and its chief role is emulsification of fats. Bile, it is not normally found in detectable quantity in urine because more than 90% of bile salt, they are actively reabsorbed. The liver normally clears about 20 grams of bile salt from the blood each day. However, when the liver is impaired or if it is damaged, the blood level of bile salt is increased. And due to liver infections like jaundice, the bile salts are produced in much larger quantity. And the common bile salt that are found are sodium glycocholate and sodium taurocholate. So students, with this, we finish all the tests for analysis of urine sample to see the presence of normal and abnormal constituents. And with this, we also finish all the working experiments in our syllabus. In the next session, we will be starting with the demonstrative experiments, which are also called as spottings.